We are continuing with evolution. We went over basics last week. This week we're going to go over a little bit more specific information. So how biological diversity evolved. So all about species. How did new species come about? We first have to know what a species is. So species is a Latin word meaning kind or appearance. The biological species concept defines a species as a group of populations whose members have the potential to interbreed with one another in nature to produce fertile offspring. So there's a lot to unpack in that definition. One of the important parts is to interbreed, meaning they can have babies together in nature, so not guided by human hands or artificially inseminated like we like to play with animals and make hybrids. So this has to happen in nature, and they have to produce fertile offspring. So the offspring have to be able to have their own offspring. So the evolving of new species according to this concept is called speciation. So when two organisms grow so far apart that they cannot interbreed or they can't produce fertile offspring anymore together, they are considered two separate species. An example is a mule. A horse and a donkey can interbreed. Their offspring is the mule, but mules are infertile. They can't have their own babies. You can't mix a mule and another mule and make mule babies. It always has to be the offspring of a horse and a donkey. So the horse and the donkey are considered separate species because their offspring are not fertile. However, several factors can differentiate a species. Morphology, which is the fancy name for just body form, what a body looks like. Physiology, the insides of something. And biochemistry, and of course, DNA sequences. DNA sequences is always the newest discovery because we've only known what DNA is actually made of for 70 years or so. Relatively new field of study. There are two types of barriers that could prevent mating or fertilization between species. Pre and post. Pre means before, post means after. This should be easy to remember. Pre-zygotic barriers prevent mating or fertilization between species. Physical isolation is the typical one. A mountain range pops up between two populations of squirrels. Those two populations of squirrels aren't going to be in physical contact with each other anymore. Eventually they grow apart. And then post-zygotic barriers happen after fertilization. That's like the mule. The hybrid, or the mix of two species, offspring is either not viable or not fertile. Like I said, horses and donkeys can mate, they can even breed, but their offspring is not fertile. This is just a handy chart. Pre-zygotic barriers is temporal isolation. Temporal means time. So those are like shifting seasons. Some animals only mate during a certain season. If the seasons shift too much, they don't get to mate anymore because they don't mate in the same season. Habitat isolation would be like that mountain range popping up. And then behavioral isolation, like, birds all have mating dances. Well, if the mating dance of one type of bird gets too far off, they're not going to mate with these types of birds anymore. And then mating attempt mechanical isolation, meaning their actual pieces don't fit together when trying to mate, or gametic isolation, that would be their gametes either don't fertilize each other or, like I said, the next generation is not fertile. Mechanisms of speciation, so how it actually happens. A key event in the potential origin of a species occurs when a population is somehow cut off from other populations of the parent species. The two different ways this can happen is either allopatric speciation due to the geographic isolation, or sympatric speciation without geographic isolation. Geographic isolation is one of those examples. A mountain range pops up, splits two populations so that they grow differently on each side of the mountain range. Or a canyon can pop up due to an earthquake, or a river can split two populations. Something having to do with geography isolating these two populations. Well, these are just pictures. Allopatric speciation occurs after geographic isolation. There's that mountain range I was talking about. And then sympatric occurs without geographic isolation. A new population just pops up in the middle of the old population. So allopatric speciation occurs with the evolution of reproduction barriers between the isolated population and its parent population. And even if the two populations should come back into contact at a later time, the reproductive barriers will keep them as a separate species. And that barrier, that river or mountain or canyon that separates the population, can do a lot of things to shift those two populations away from each other and becoming two separate species. $251 million years ago, all continents were connected as Pangea. And the breakup of Pangea is an extreme example of allopatric speciation. All of those animals that were on one big continent together suddenly got separated into a lot of different places. We will come back to Pangea in a few minutes. Sympatric speciation occurs in populations in the same geographic area, so they're not isolated from each other. An accident during cell division that results in an extra set of chromosomes is a common route to sympatric speciation in plants. So all of a sudden, a plant population in the middle of a forest somehow mutates to have an extra chromosome. It can no longer mate with its parent population of trees around it. Trees they do a lot of self-fertilization anyways. It can only mate basically with itself. And that new population starts to grow within the old one. Geologic time. Geologists have established a geologic time scale that divides Earth's history into a consistent sequence of geologic periods. We've all heard of Jurassic Park. Jurassic was an actual period in time on the geologic time scale. And a lot of dinosaurs lived during this time. So that's where that movie got its name. They could have called it Triassic Park or Cretaceous Park too, but Jurassic just went better. Here's an overview of all the different time periods and what animals appeared during that time period. Fossils are reliable chronologic records. Fossils are reliable chronological records only if we can determine their ages. And we do this a couple different ways. We use relative dating. We can determine the relative age of fossils. revealing the order in which groups of species evolved. Oldest fossils can always be found on the bottom. Younger species can be found on the top. And then sometimes you have a little cut going across all of them. That would be the youngest. The absolute age of fossils requires other methods, such as radiometric or radiocarbon dating, where they actually measure how much carbon is in a fossil and how old that carbon is based on its half-life. Plate tectonics and macroevolution. The continents are not locked in place. Continents drift about the Earth's surface on plates of crust, floating on a flexible layer of hot underlying material called the mantle. If you've taken any kind of earth science class, this is what you learn about. And the plates shift against each other. This is also what causes earthquakes. About 250 million years ago, the plate movements formed the supercontinent Pangaea. The total amount of shoreline was reduced. The ocean basins increased in depth. Sea levels dropped. And the dry continental interior increased in size. And because of this, because there's a lot of species that no longer had access to water, many extinctions occurred. About 180 million years ago, Pangaea began to break up. Large continents drifted increasingly apart, climates changed, and the organisms of different biogeographic realms diverged. Now, all of a sudden, organisms had access to water again. So a lot of water creatures could crawl up onto the earth. Plate tectonics helps to explain why Mesozoic reptiles in Ghana in West Africa and Brazil look so similar, because they used to live very closely on that big continent. And then how marsupials were free to evolve in isolation in Australia. The ancestor, we saw a video last week, or not last week, on Monday. The ancestor of the marsupials started somewhere in China, made its way all the way to the southern tip of Pangaea, which eventually became Australia. And it evolved into many different marsupials from there. But when Australia broke off, they had nowhere to go, so they only had Australia to evolve in. Classifying the diversity of life. Taxonomy is the identification, naming, and classification of species. Scientific names ease communication by unambiguously identifying organisms. Unambiguously means clearly. And making it easier to recognize the discovery of new species. And they're all Latin names. And no matter what language you speak, if you're a scientist, you always use the Latin names for species. It makes it easy to work with other scientists. You know exactly which organism you're talking about because you use the same name for it. Some basics. Carolus Linnaeus proposed the current taxonomic system based upon a two-part name for each species and a hierarchical classification of species into broader groups of organisms. Side note, Linnaeus's work on the classification of man forms one of the 18th century roots of modern scientific racism. And there's more information about that if you'd like to dive into that. It's just kind of an interesting side note. And the Linnaean Society actually recognizes that and is doing all they can to rectify it. But he was the first one to put animals, plants, minerals into actual groups and say, these look like these, so they should be one group. These are all plants, so they should be another group. Each species is assigned a two-part Latinized binomial name, consisting of a genus and a unique name for each species. The scientific name for humans is Homo sapiens. So it's two parts, it's italicized, it's given a Latin ending, the first letter of the genus is capitalized. Like I said, it doesn't matter what languages scientists speak, everyone uses the Latin names for the organisms. The taxonomic hierarchy extends to progressively broader categories of classification, from genus to family, order, class, phylum, kingdom, and domain. You might have learned these in another science class once upon a time. We don't really talk about them in regular biology, but I vaguely remember learning them in like elementary school or junior high. So this is an example. This is a leopard. Its species is the Panthera pardus. Genus is Panthera. Family, order, Carnivora, meaning it's a carnivore. Class, Mammalia, meaning it's a mammal. Phylum, Chordata, meaning it has a backbone. Kingdom, Animalia, it's an animal. And domain, Eukarya, because it's a eukaryote. It has eukaryotic cells. So each group gets progressively bigger. Species that are closely related are placed into the same genus. Example, the genus Homo, which is us, our genus, includes a number of different hominids. Homo sapiens is us. Right before us, or the Homo neanderthalensis, the Neanderthals. Homo heidelbergensis, Homo erectus, Homo habilis, Homo ergaster. We are all closely related, so we are placed in the same genus, but we are not the same species, which is why the second name, sapiens, neanderthalensis, those are different. Biologists use phylogenetic trees to depict hypotheses about the evolutionary history of species and reflect the hierarchical classification of groups nested within more inclusive groups. So this is just one very small branch of the tree of life. You can see on the bottom the order Carnivora. You can think of a lot of carnivores. And there's a bunch of examples of carnivores. The left branch is the dog family, Canidae. And the right branch is the cat family, Felidae. Either way, they're still all carnivores. And then it just gets more and more specific till you have the specific cheetah, tiger, lion, puma, cat. And they all have their own specific genus and species. This is what the whole tree looks like. And even one on the top left, bacteria, archaea, eukaryota, they're still only part of the tree. But it can look like branches, it can have actual pictures of animals, or it can look circular, like the one on the bottom. Homology and analogy. Homologous structures reflect variations of a common ancestral plan and are one of the best sources of information used to develop the phylogenetic trees and classify organisms according to their evolutionary history. So organisms that have the same structures are typically related. Not all the time. Sometimes they're just analogous structures. They're the same structure in organisms, but they did not come from a common ancestor. So these are not related. An example is wings. Birds have wings, insects have wings, and bats have wings. But they are very, very far apart on the phylogenetic tree. They are not very closely related. The wings of birds, if you look at an X-ray, are attached to their arms. If you look at a bat, the wings of bats are actually attached to their fingers, which we'll see in a minute. And insect wings are completely different from both of those. They do the same thing in all three organisms. They help it fly, but they evolved independently of one another. Molecular biology. Molecular systematics compares nucleotide and amino acid sequences between organisms and can reveal evolutionary relationships. Like I said, this is one of the newer fields of study because we find out so much about DNA every year. The more recently two species have branched from a common ancestor, the more similar their nucleotide and amino acid sequences should be. Some fossils are preserved in such a way that DNA fragments can be extracted for comparison with living organisms. We talked a little bit about this last chapter. Us and chimpanzees shared like 98.7% of our DNA if you compare it. But that little bit that's different, there's specific gene sequences that let us know how we're different. One gene sequence is for a bigger brain. We have a gene sequence for a bigger brain. We have a gene sequence to have our mouths form different ways so we can form actual words. That is found in our DNA and is different from chimpanzees. They are a work in progress. Phylogenetic trees are hypotheses about evolutionary history. Linnaeus divided all known forms of life between the plant and animal kingdoms. He didn't know any better back in the 1800s, 1800s. And this two-kingdom system prevailed in biology for over 200 years. Everybody thought everything was just either plants or animals. With the invention of things like the microscope, we had to readjust our classification system. In the mid-1900s, the two-kingdom system was replaced by a five-kingdom system. We placed all prokaryotes in one kingdom and then divided the eukaryotes among four other kingdoms. And then more tools were discovered, and we found more information about the natural world, so we had to overhaul the classification system again. In the late 1900s, molecular DNA studies and cladistics led to the development of a three-domain system, recognizing two domains of prokaryotes, bacteria and archaea, and they put all the eukaryotes into the other domain. So previously, you just had the kingdoms, and with the discovery of DNA and all that went with it, they went higher than kingdom into domains.